Okay, so today I'm going to talk about the extracellular enzymes, intracellular bioproducts, and exopolymers, which we call them as unlocking the microbial biotechnology. So basically, this aspect of microbial technology or microbiology with the industrial application of enzymes unlock the microbial biotechnology and explore more of like biotechnology area where we can explore the bioproducts utilized by the microbes and taken out from the microorganisms and for the benefit of the mankind. Now let's talk about the introduction to microbial bioproduct. Already we have talked about many bioproduct and metabolites, secondary metabolites and primary metabolites in the earlier lectures. So these secondary and primary metabolites are also sort of bioproduct from the microorganism, but these bioproduct can be extracted out from these microbes and microorganisms, which can be utilized by the mankind. Now the microbial factory is considered as a tiny cell which has a massive impact on the whole economy, whole business, and whole the use of these microorganisms. So the microorganisms represent the nature's most versatile biochemical factories capable of producing an extraordinary array of molecules that have transformed multiple industries. Now these microscopic powerhouses synthesize enzymes, polymers and metabolites with remarkable efficiency and specificity. Now the biotechnological applications of the microbial product span across pharmaceutical and manufacturing, food processing, agriculture enhancement and environmental remediations. Right. Now Indian biotechnology sectors has particularly benefited from these microbial innovations, which produce a lot of bioproducts through these and give significant input to the microbial or economic growth of the industry. Now the pharmaceutical industries, uh, among the pharmaceutical industries, the product which are being produced are major product are antibiotics from the bacteria, fungi, vaccines from from different ways, synthetic vaccine as well as the microbial vaccines. So the attenuated vaccines and the vaccines produced through microorganisms, and the therapeutic proteins, which are very, very important. So these therapeutic proteins are produced using these microbes or microbial industries by recombinant DNA technology, or the protein itself can be purified through wholesale extract of these microbial microorganisms or microbial worlds. Now in the food processing industry, the role of these microorganisms is very significant, very significant. Enzymes for the brewing, baking and preservations, all these things, these products can be used for the production of the better quality food and preservation of the foods, and the foods which can have give better growth economy in the future as well as in the current situation. The environmental applications of these microbial world or microorganisms are well documented for remediations, that is bioremediations, that is use of these microorganisms to remediate or to disinfect the various pollutions, treatment of plants, wastewater plant and several other things. Now defining the key players, what are the key players in the bioproduct production? So extracellular enzymes, intracellular products and exopolymers. These are the main key players which are involved in the industries and the application of these microorganisms for the industrial production. The extracellular enzymes, they are catalytic proteins which can catalyze the several reactions, biochemical reaction in the cell, and these are secreted by the microorganisms. So they have their role in the microorganisms, but they can also be extracted, can be purified from the microorganisms for reactions outside to break down the complex substrates in their external environment. Now these enzymes enable microbes to access nutrients whilst providing the valuable industrial applications. Right. Now the intracellular product, the valuable compounds accumulated within the microbial cell, including the antibiotics, vitamins, some pigments, dyes and bioplastic precursors, and these products require cell disruption for the extraction and purification. So the extracellular enzymes, without disrupting the cell, by growing the cells, you can extract it out. Whereas the intracellular product, you need to lyse, you need to disrupt these cells in order to get these products out, like antibiotics, vitamins, some dyes and pigments, bioplastic precursors. Many times these intracellular products can also be used as a recombinant proteins, a recombinant gene product which can easily be identified. For example, in case of bioplastic precursors, polyhydroxybutyrate, right, which can have a property of the biodegradable nature. So we can produce these through microbial world, microorganisms. I'm using the word microbial word means like the holistic view of the microorganisms. That could have like whole fungi, bacteria, different kinds of bacteria, different kind of fungi. So we call the whole microbial world. So the whole microbial world means like using multiple microorganisms, diversified microorganisms can be used for this purpose. Now the exopolymers, high molecular weight polymeric substances secreted into the surrounding environment, and these complexes molecules serve protective function whilst offering diverse industrial applications. So this is how it works, exopolymers. And sometimes exopolymers are also intrinsic property of the microorganisms, which can also protect the microorganisms from the various defense system. Extracellular enzymes, the nature's catalysis beyond the cell. Exploring the production, applications, and biotechnological significance of the enzymes secreted by the microorganisms. What are extracellular enzymes? Extracellular enzymes are sophisticated protein catalysts secreted by the microorganisms to degrade the complex substances or substrates as to use for the production of various bioproducts in the external environment. And these enzymes enable microbes to break down the polymeric substances into small absorbable units. Now, this basically means like how these molecules these substrates can be used for the production of the byproduct using the enzymes produced by the cells. So these extracellular enzymes can be utilized for various industrial applications as well. Now, the secretion process involves the sophisticated cellular machinery, including the signal peptide, transport system, regulatory mechanism that ensure the proper enzyme folding and activation outside the cell. Now, it means the internal mechanism of the cellular internal mechanisms is also involved in making the enzyme more functional. So we call folding here means like because enzymes is a sort of a protein, so these protein need to be functionally active. In order to make the proteins more functionally active, what is being required is the proper folding of these enzymes. Now, industrial powerhouse, we call enzymes applications. Why we call it as a powerhouse? Because it produces energy, and how the energy is produced through these enzymes, various metabolic reactions and metabolic processes, we call them physiological reactions, have been taking place in bacteria, in fungi, in various microorganisms, and these microorganisms use these enzymes as to convert into the various substrates or substances or substrates into the product. And so, for example, and these products are used in various purposes. So, the global enzymes market values like billions of dollars of these continuous expanding rapidly, with the Indian companies playing an increasingly important role. So, having that information, having that knowledge of the enzymes used for the various extracellular enzymes used for various purposes are very, very significant and very important from the industrial applications, and of course that grows the economy of the country and the global economy. Taking some examples like in case of textile industry, which is one of the biggest industry of the world and the country as well, where the cellulase enzymes are important, where it provides the biopolishing of fabric, and biopolishing which has better effect on the cotton fabrics. They can make more like soft textures of these cottons, and it improves the appearance of these materials, the cloth and the cotton materials, thread materials, and it could have different fine textures can be produced through these cellulase enzymes. and it also reduces the environmental impact compared to the traditional chemical treatment. So, comparing to the chemical treatment of this fabric industry, which can also produce the pollutant as effluent of these industries, and that may lead to the problem and disaster in the water pollutions, environmental pollutions, and also these are the toxins, and there may be some carcinogenic compounds, and these carcinogenic compounds can be harmful for the uh, manpower which are being used to handle these textile industries, or they work in the textile industry. So all these things can be overcome. These risks can be overcome using the cellulase enzyme. Now, the food processing, like amylase, enhances the bread texture and extends shelf life, whilst also the facilitating the beer production by converting the starch into the fermentable sugar during the brewing process. So, the food industry is again a big industry. Again, it's the global industry where the amylase enzymes. So, if you producing the amylase through microbial world, and if you use amylase to enhance the bread textures and extend the shelf life of these food products, different food products, uh, converting the starch into the fermentable sugar molecules for various uses, for energy productions and for various other purposes in the food industry during brewing process, then you can protect the food industry from use of the various chemicals, various synthetic molecules or synthetic enzymes which may be harmful for the food industry as a food additive. So this is again one of the advantage of these enzymes applications. Now, the detergent manufacturing is again a very important area where the protease and lipases are enzymes which are used for effectively to remove the protein and fat-based stains, and these stains at a lower temperature enable the energy efficient washing whilst they are maintaining the superior cleaning performance of the cloth or of the fabric. So, the fabric quality remain maintained without damaging it. These detergents work well if they use the proteases and lipases. I mean, they can easily remove these stains of proteins and lipids, the organic compounds. Now, the last but not the least, the biofuel production, which is again a big challenge before the whole world because there's a crisis in the fossil fuel. So now we have to look into the alternative approach, that is biofuel productions. Now, biofuel production is totally based on the biological molecules or bioproduct, where like a plant and various microorganisms, and the cellulase break down the lignocellulosic biomasses of these raw materials of the biofuel into the fermentable sugar molecule, and this sugar enable the sustainable production of a second generation bioethanol, right? And these bioethanol from agriculture waste materials can be utilized. Now, some molasses and some other materials is also used as a substrate for production of these biofuels using these enzymes which process these product into better way and can produce high quality biofuels. Now, the case study, one of the case study in cellulase from Trichoderma resci. Now, Trichoderma resci is one of the species which produce the cellulase enzymes, and it has a revolutionary impact on the bioethanol. So, Trichoderma resci represents a landmark success in the microbial enzyme, right? And the production of enzyme is also increased, demonstrating the exceptional cellulase activity. It means the cellulase activity may also increase the efficiency of these cellulase enzymes to convert these cellulose into the sugar and other molecules for the purpose of this production of ethanol as a substrate or as a primary product. So that has transformed the renewable energy landscape. So the renewable energy can be transformed into like using these biofuels. So we can produce a lot of energy from these biofuel productions, and the biofuel productions can also be controlled or regulated, at the cost of like manpower, at the cost of like techniques being used for these enzymes production. So the filamentous fungus produces a complete cellulase enzyme. and the system capable of efficiently hydrolyzing crystalline cellulose. So cellulose is completely hydrolyzed in well, making it industry and standard of bioethanol production. So they can produce the precursors. Now the high specific activity against cellulose substrates, uh, because of the high specific activity against cellulose substrate, it has excellent thermal stability for industrial processes and scalable fermentation production methods, cost-effective enzyme recovery and purification. So, all these features of these fungus cellulase is very important for production of the high quality biofuels or bioethanol. Now, the enzyme production, fermentation and optimization, sorry. Uh, Now, one thing to be taking into care here, whenever you talk about the production of any enzyme or through fermentation or through any mean, the optimization is very important. Optimization depends on many factors, right? Optimization depends on various approaches, right? And you can improve the yield of these product through fermentation by optimizing these things, like. So, what are the different ways where you can have like better optimize in order to produce high quality and high quantity product? So, the strain selection and development. Now, strains are very important. What are the strains you are using? Are these strains are good quality which can produce the better quality and more quantity of the biofuels or of the substrate to convert into the product? So, you have to identify the high producing microbial strains, and if not, then you can have some mutations, and you can also generate by spontaneous mutation and selection of these mutations through different means. Now, you have to screen the different mutants, and you can genetically modify these mutants of these fungus, of these microorganisms, and you can also produce through some techniques by side-directed mutagenesis and to enhance the enzyme expression level. So, the idea is that how the enzyme expression level is increased, and when the enzyme expression level is increased, then you can have a better conversion of the substrate into the product. So, strain selection is very important. So, if you have a good strain selections, high quality strain selection, or you modify these strains through mutations or through modification, then you can have better quality biofuel through fermentation, and this is one of the way of optimization of fermented product. Then medium optimization, for example, what medium you are using, what are the nutrients you are providing, are these rich in nutrients for giving the fermentation process better and high quality yield, what are the optimal carbon and nitrogen level in this, what are the energy sources, what are the trace elements, what are the growth factors, are the maximum enzymes productions can be attained on these media or not. So, that has to be looked into, and if you can modify it, you can optimize this, you can have a better yield of your product. Then process parameter control, again you have to have some physical parameter which are required for the growth of the cell under a specific media, under specific strain selection. You have to optimize the physical parameter. For example, what is the pH, what is the temperature, what is the dissolved oxygen, or what is the CO2 concentration if it is something gram positive or anaerobic condition. So, what kind of a condition you are required for this fermentation process, exonic or anaxonic, or anaerobic cells or anaerobic cells. So, on the basis of the strain selection and the media, you can also optimize the physical parameters or the parameter that can control and regulate the continuous quality and yield of this process. Then you have a downstream processing. Now, in downstream processing, we have implementing efficient separation, purification, and concentration techniques to isolate high quality enzymes suitable for industrial applications. Now, implementing the efficient separation, purification, and concentration techniques to isolate high purity enzymes. Now, this is very important. How once you, for example, if you have some recombinant molecule and you have to produce the protein or enzyme through the recombinant molecule, then what are the condition to purify is to be identified, and that should be high purity enzyme conditions where you can have high 99.9% purified enzymes can be isolated. And therefore, for industrial application, the downstream processing is very, very important to get the best quality and high quantity, good yield of these products. Now, the biotechnologically important intracellular product. What are the different intracellular products we have? Again, intracellular products are basically identified through, for example, antibiotics, which are produced from intracellularly. Then you have natural pigments, vitamins, and bioplastic precursors. So, intracellular products represent some of the products which are most valuable from biotechnological perspectives and for the mankind, and these products often require the sophisticated extraction method, but offer exceptional purity and biological activity. So, having intact your biological activity and purity of these compound, what you have to do, you have to use very sophisticated techniques to get these intracellular product out. So, what is most important is the purity of these product. For example, if you are using the antibiotics, so antibiotics, since it is a life-saving molecule, life-saving compounds, for example, penicillin, streptomycin, tetracycline, they are all produced from the bacteria. So, from the fungus. Now, when the fungus is these are isolated from the fungus, then fungus byproducts and some traces may also come along with the antibiotics. So, you have to be careful how single molecule and compound can be well purified from these microbial system or microorganisms, so that you can have very purified kind of antibiotics. Now, the natural pigments is again like carotenoids, and then you have astaxanthin, then you have other valuable molecules, valuable colorant, antioxidant molecules with the properties, and these colors can be used for various purposes. Various dyes and various other things can be synthesized through these pigments. Essential vitamins are very, very important because these vitamins are again for the living system, to make the living system intact. Like vitamin B12, they have different functions, riboflavins and other crucial multivitamins, are B complex and so on. They all can be produced from the microorganisms, and they are very crucial microbial nutritional supplements. So that need to be very purified in many ways. And the bioplastic precursors, again, polyhydroxy butyrate, polyhydroxy alkylamide is one of the examples, polyhydroxy butyrate, and the biodegradable polymers building blocks. So these again can be produced through various precursors of these intracellular product and need to be purified. Antibiotics, microbial defense turned medicines. So, for example, these antibiotics are very, very important, as we already talked about these earlier lectures, that its discovery in 1928, then discovery of streptomycin in 1940, and then tetracycline in 1950. Then presently Indian production is like seized, and because of some reason, and pharma industries are not focusing on this production of these antibiotics, but they are more focusing on some alternative medicines and other medicines, and they are just making these different antibiotics in different forms through chemically synthesized molecule using the same nucleus of these old discovered antibiotics like penicillin, streptomycin, tetracycline, of different groups like aminoglycosides, beta-lactams, and fluoroquinolone, quinolone groups of antibiotics. Now, so these all antibiotics we discovered through these bacteria are important and good for the health, and in the health system they are very, very significant. Microbial pigments and vitamins. So natural colorant revolutions is very significant from the economic growth perspective. And for example, consumers demand for the natural food additive. So because the chemical colorant is basically maybe toxic, maybe carcinogenic. So natural food additives are required where they can add these food colors. They can be derived from the various microbial pigments, and these compounds offer superior stability, safety, and functionally compared to the synthetic alternatives. Carotenoids are from the algae basically, and bacteria, cyanobacteria, they are produced, and they are vibrant colors whilst delivering the antioxidant benefits. Then we have we can also use prodiagnosin produced by the Serratia mercescens, and then often striking for the red pigment with the potential anti-cancer properties. So these compounds can also be used for various purposes in medicine, that is medical industry and food industry. Beta carotene production, it's a provitamin A precursors from the Dunaliella salina, and then we also have another species of Blackacilia, then Trispora, for the nutritional supplements and food additive as a color application in the food industry. Similarly, vitamin B12 is produced from the Propionibacterium species, produced cyanobacteria or cyanocobalamin through the complex biosynthetic pathways. So the cyanocobalamin is produced from the Propionibacterium species in order to produce the best quality vitamin B12, which is essential for various vegetarians' dietary supplements for health issues and various strengthening of various function in the health system. Then we have extraxanthine production is again a pigment with a powerful antioxidant from the Hematococcus pluvialis, and this commanding the premium prices in the nutraceutical. So besides food industry, the nutraceutical industries are also growing because of these microbial intracellular products, and aquaculture market is also one of the area or one of the domain where these can be used widely. Bioplastic is polyhydroxy alkyl PHAs, are one of the examples is polyhydroxybutyrate. These PHAs represent the promising solution for the global plastic pollution crisis, where these are biodegradable plastic. So bacterial accumulation, like we have Cucurbitacus necator and other bacteria like accumulate in PHAs as energy storage granules, and when the carbon is abundant but other nutrients are limited. So when under the limited conditions of the different nutrients where the other nutrients are limited, the abundance of these accumulation of these molecules takes place, and that can be utilized later on by isolating from these bacterium as a PHA. Polymer extraction, cell disruption, so you have to lyse the cells to extract. Solvent extraction techniques are used, which can recover these granules, and which are the purified form of this plastic and can be processed into several plastic products later on after purification. Then manufacturing of these products, PHA can be modeled and then extruded and processed using the conventional plastic manufacturing equipment to produce the biodegradable alternative. So this is one of the advantage of the microorganisms. The extractions and purification challenges. What are the different extraction and purification challenges? There are certain challenges and which can be handled through several techniques and can be optimization process in order to get the purified intracellular and extracellular product. Now, one of the challenges in purification is mechanical disruption. So high pressure homogenization, ultrasonications, and bread milling provided the efficient cell breaking and maintaining the product integrity. So this is need to be has to be developed. So I mean, without these facilities or without these technologies, you cannot have mechanical disruption. Now, as far as the chemical lysis is concerned, the detergents, organic solvents, and alkaline treatments selectively permeabilize cell membrane for the gentle product release, right? So this is how chemical lysis takes place. The downstream processing, chromatographic separation, crystallization, and concentration techniques and achieve pharmaceutical grade product purity. Now, enzymatic treatment, lysozymes and other cell wall degradable enzymes which can degrade provide the selective cell disruption with the minimum product degradation process. So ultimately, how to extract these intracellular product and purify these product is challenging task. So we can use different methods, mechanical disruptions, chemical lysis, and downstream processing and enzymatic treatment in order to extract as well as to purify these process. So now finally we conclude that intracellular and these extracellular product which are produced by the bacteria are useful in various industries, food industry, medical industry, pharma industry, and it can also be used for various other industries where, like fabric industry, for for various purposes of making the product in a better, economically good and high quantity and good quality product can be produced. So this is all about this topic. So now thanks a lot, and we'll see you in next lecture soon.